Hello, everyone. Welcome to today's live broadcast, Understanding SingleParticle ICP MS in Nanotoxicology and Ecotoxicology. I'm Laura Bush, the Editorial Director of Spectroscopy, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by Spectroscopy and sponsored by PerkinElmer. PerkinElmer is a global technology leader focused on improving the health and safety of people and the environment. The company provides solutions that help customers perform advanced detection and analysis across environmental, food, and industrial applications. Their instruments, software, and services enable analytical labs to detect, monitor, and manage contaminants and toxic chemicals that affect the safety of our environment. We have a few housekeeping announcements before we get started. The webcast is designed to be interactive, so we encourage you to ask questions. You can submit questions at any time by typing them in the Q&A box, and you can find that on the left-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the webcast. If you have any technical problems viewing or hearing the presentation, please click on the question mark Help widget in the dock at the bottom of your window. I would now like to introduce today's speakers. We're very pleased to be joined today by Chadi Stephan, Jamie Lead, and Ruth Merrifield. Dr. Chadi Stephan holds a PhD in analytical chemistry from the University of Montreal, and he worked as a project manager for QSAR Risk Assessment Services before he joined PerkinElmer as the inorganic product specialist supporting various elemental analysis platforms. He currently manages the development of various nanotechnology applications that focus on measurement techniques for nano-object characterization. He's a thought leader in the nanometrology space with more than 20 peer-reviewed papers and book chapters. Professor Jamie Lead is an endowed professor and the director of the Smart State Center for Environmental Nanoscience and Risk at the University of South Carolina. His research aims at understanding nanoscale phenomena in the environment, and he is interested in investigating natural nanomaterials, manufactured nanomaterials, and their interaction. Professor Lead has published more than 160 papers and edited 3 books, and he holds 3 patents. He's an honorary professor and the former director of the Facility for Environmental Nanoscience Analysis and Characterization at the University of Birmingham in the UK. He is also a fellow of the Royal Society of Chemistry and the International Union of Pure and Applied Chemistry. He's the editor-in-chief of the journal Nano Impact. Ruth Merrifield works as a research assistant professor in the Smart State Center for Environmental Nanoscience and Risk at the University of South Carolina. Her research focuses on characterizing nanomaterials with environmental systems and developing new tools to assist in the understanding of these processes. She earned her PhD in nanoscale physics at the University of Birmingham, followed by a postdoctoral position in nano-ecotoxicology at the same university, combining her interest in the environment with her love of physics. Thank you all for joining us today. Chadi, you have the floor.
Thank you, Laura. Good morning, good evening, or good afternoon from wherever you're joining us. On today's webinar, I have as Laura stated, we have the luxury of having Dr. Lead and Dr. Merrifield joining us today, talking about the ecotoxicology and the nanotoxicology of nanomaterials. For today's webinar, I'm going to go and cover a little bit of introduction around the characterization technique of nanomaterials, and I'm going to, after that, give the floor to Dr. Lead, who's going to talk about the Center of Environmental Nanoscience and Risk. And after that, we're going to have Ruth or Dr. Merrifield discuss the SingleParticle ICP MS and its usability and applicability in looking at the transformation of various nanomaterials and their implication into the nanotoxicology and ecotoxicology space. So as we all know, nanomaterial characterization is complex. Nanomaterials themselves are complex. They have unique characteristics. We, as a scientific community, we probably lack a little bit of techniques that allows us to better understand and uncover the mechanisms or the characteristic of such materials. on that, we've started about an effort at the ISO about 3 years ago, looking at the various techniques that we have currently as scientists to look at nanomaterial characterization. And I will refer to that as an ISO 18196.2, in which we are talking about 30 plus techniques that could help us better understand nanomaterial characterization. PerkinElmer and being part of the organization have been focusing the PerkinElmer portfolio around the nanomaterial characterization. And early on, we've worked on developed the SingleParticle ICP MS that allows us to look at the composition of nanomaterials and mainly track nanomaterials or nanomaterials transformation at the environmental level. But to that, PerkinElmer has a whole suite of analytical technique that helps better understand and characterize nanomaterials, either if they are embedded into what we call them advanced polymer or advanced materials. So basically, we're looking at various material characterization techniques such as DSC, differential scanning calorimetry, and others. We've also very much on the lead in looking at hyphenation techniques. And hyphenation techniques are basically putting various detectors in line together towards achieving the measurement that we need. And one of the most valuable technique for nanomaterials is a TGIR GCMS, in which we look at the evolved gas that leaves nanomaterials and nanostructure and combined with the precise measurement of the gas chromatography mass spectrometry. And that kind of gives you the big idea about what we've been trying to do at PerkinElmer towards the improve or provide tools for nano researcher to improve their research. Today, we're going to be talking about the environmental implication, the environmental toxicology of nanomaterials. How can we track them? And for that, I put this diagram up here looking at the environmental cycle of engineered nanomaterial, going from the end of life, being released into a wastewater treatment plant, for example, and all the process that takes it into the wastewater treatment plant towards having nanomaterials makes it to the effluent, and then the environmental process that takes in nature regular surface water or a river or a stream, and basically as well we're probably going to have some nanomaterials or engineered nanomaterials entering the biosolids or ending up in the biosolids and then going to be entering the soil. What are the available techniques that allows us to track what happened to nanomaterials into the various spaces? To summarize that, we're looking at the environmental transformation of engineered nanomaterials. So what's going to happen to those nanomaterials? In that diagram, we're talking about 4 different types of transformation. Nanomaterials could go through a dissolution or could have some reaction happening on their surface. They could aggregate, they could dissolve, they can end up at the bottom of the river or on the bed of the river. They could have some absorption and desorption. They could dissolve and reprecipitate, or nothing could happen to them as like type 1. They're just going to be persistent and keep on going. So all those transformation needed an analytical instrument or needed a technique, let's put it this way, it's a technique that allows us to track the transformation of nanomaterials at the environmental level. And for that, as we're going to hear from Dr. Merrifield in this webinar, how SingleParticle ICP MS is playing a key important role in understanding the transformation, which is going to impact the toxicology and the ecotoxicology of nanomaterials or their effect on the environment. With that, I'm going to give the floor to Dr. Lee, who's going to sit from here. Thank you.
Thank you, Chadi. Hello, everybody. I'm Jamie Lead, the director of the Center for Environmental Nanoscience and Risk. And I'm going to talk a little bit about the center itself and give you some background as to what we do, but also set the scene a little bit for Dr. Merrifield's talk on SingleParticle ICP MS, particularly why we're interested in looking at the physical and chemical characterization and properties of nanomaterials, particularly here in relation to nanotoxicology and nanoecotoxicology. But this is an area that's of importance more widely for areas such as nanomedicine, for the safe and sustainable use of nanotechnologies, and for environmental behavior more generally. The Center for Environmental Nanoscience and Risk is a collaboration between state, university, and private sources to understand environmental nanoscience and risk more broadly. And we're well set up. You can see in the picture below there are a number of students and staff and faculty as part of the center here. We're well set up for synthesis, characterization of nanoparticles, and associated areas of science, including toxicology and related issues. And we're really interested in two specific areas. So we have two specific aims. One is the improved understanding of environmental implications of nanoscience, and that includes ecotoxicology and toxicology, because the environment is an important exposure route to humans. And that will be what I'm talking about throughout most of my talk before handing off to Dr. Merrifield. The second area is in developing environmentally friendly applications of nanoscience and nanotechnology. And here we're focusing on specifically uses in the environment of nanoscience, and also uses with a low environmental footprint, so minimizing use of toxic materials, minimizing energy and material inputs to make nanomaterials. And just to give you a couple of examples of the work we're doing here, the first one at the top is work with oil remediation. So we're using polymer-coated magnetite nanoparticles for oil remediation in natural systems. And we're particularly focused on this area because it's an effective, cheap way of remediating oil in natural systems. It works under natural conditions, which are very challenging. They're easy to make and cheap to make. They have a low toxicity. And also, interestingly, we just found that they stimulate the natural remediation capacity of environmental systems, particularly the bacterial capacity of these systems. And there below you can see an example of the work we do with an oil-water mixture on the left-hand side of the image. And then as we move through various exposure times, up to about 40 minutes, we can essentially remove all of the oil from an oil-water mixture. And essentially what's happening here is that the oil, driven by the hydrophobic effect, is binding to the polymers on the magnetite. And then we can use magnets to easily separate the particles with the oil from the water, and essentially over 30 minutes to an hour remove all oil from an oil-water mixture. The second area is related to our work with toxicology, looking at nanohybrids. So nanohybrids are combinations of different types of nanomaterials. So for instance, we're looking at isotopically labeled metals, mixtures of metals, and in other projects we're looking at metal carbon-based hybrids. And these materials may have different properties from the nanomaterials that they're made from. And that's one important question in the field of an emerging question in the field of toxicology and in environmental nanoscience. With these nanohybrids, are they persistent for long enough periods to have an effect as a hybrid, or do they dissociate into their component parts and can be treated as the original nanomaterials? And we're actually using the hybrids on the right, which are isotopically labeled core shell particles, to try and understand how nanoparticles are bioavailable, how they bioaccumulate in organisms, whether as a particle or do the particles dissolve and the ions are then uptaken. And we do this by being able to separate the core material, which is one isotope, from the outer layer, in this case silver 109 in the diagram, which is partially soluble, and it's separated by an insoluble layer of gold or similar material. And using this combined with ICP MS and SingleParticle ICP MS and other techniques, we can hope to answer that question of whether bioavailability is driven by direct particle uptake or is driven by dissolution followed by uptake of the ion, the silver ion in this case. So there are clear links between these two aims, particularly in that case where we're trying to use novel, environmentally friendly nanomaterials to answer questions that are important in nanotoxicology. So it's clear to anyone working in the field that the physical and chemical properties of nanoparticles will affect their bioavailability, their toxicity, environmental transport, and transformations, which I'll come onto in a minute. And so we need to know all about these physical chemical properties because they have these significant effects on these processes. And we're talking about things like composition, and that's what most people think of when they differentiate nanoparticles from each other. So that you can have gold nanoparticles or silver nanoparticles or carbon-based nanoparticles. But there are a number of other properties of the nanoparticles which are important, some of which are listed on this slide. Things like size of the particles, which can vary in the traditional definition between 1 and 100 nanometers. Coatings, which can be either the small ions, small organic ions, or polymers or other materials. The shape has been shown to affect some of these properties, crystal structure, and a number of other physicochemical properties. To the extent that there are effectively innumerable different types of nanoparticles, if we look at different combinations of all of these physical-chemical properties, we're not dealing with a single material or a single type of material. We're dealing with a whole range of different materials, some of which can be highly toxic and some of which can be non-toxic and inert. An example of that is given here from a publication of ours from 2013, where we synthesized silver nanoparticles and exposed them to Daphnia magnia, an invertebrate test model used to understand toxicity. And here we exposed in traditional iso media, which is a high ionic strength, high salt concentration media, and they aggregated, as in the TEM on the right-hand side. So you get large aggregates of these originally small particles. On the left-hand side, we see the particles, a TEM image of the particles which are exposed to the same iso media but diluted by a factor of 10. So the salt concentration causes aggregation at high concentrations, but at low enough concentrations, these particles are slightly changed from the original, but they're still dispersed. And so you get different types of the original nanoparticle that the organisms see. And on the right, we have the outcome of that. We have a dose-response relationship with mortality of the Daphnia on the y-axis and the exposure concentration on the x-axis. And the blue line is the diluted material. So the individual dispersed particles are diluted and, the media is diluted. The particles are individual dispersed particles. And what you see there is a relatively toxic material with an LC50 of a few micrograms per liter. LC50 is the lethal concentration for 50% of the population. But when we have it in the concentrated media and the particles aggregated, the dose-response curve is shifted to the right by an order of magnitude. So the aggregation of these particles into large aggregates of hundreds of nanometers changes the toxicity by about tenfold. So it's the same original material in a different form has a huge effect on the toxicity. That's one example of transformations of nanoparticles that go on, and they occur in complex media. And particularly of interest today is toxicology exposure media, as I presented just now, and environmental media. So when particles are exposed to these complex systems containing different types of salt, or natural organic matter from the environment, protein, vitamins, antibiotics, and other materials, they can transform quite substantially. And at the bottom of the slide, you see another example of that with different types of nanoparticles exposed to different systems. And we have here, again, silver particles, but with different types of coating and exposed to different types of systems. And these are modified, again, daphnia media, daphnia exposure media, where they've been modified by removing or adding different types of salts, keeping the salt concentration the same. And I won't go through all the details here, but what you can see on the right-hand side is substantial transformations. You can see the UV vis data at the bottom right, which shows shoulders from the original 400 nanometers, showing aggregation and shape change. And on the left-hand side, a different polymer coating shows losses of the material, but no real changes in shape and no real aggregation. So it's related not just to, these transformations relate not just to the exposure media and the nature of that media, but also the nature of the particle, which can be persistent or can be readily changed into something different. So we're interested in, and the ICP MS can help us with, understanding dose and exposure measurement. And roughly, we can say that the concentration in the exposure media is related to the exposure, and the amount in the cell, or the concentration in the cell of the organism is related to the dose. And an interesting question that comes up in this area is how do we measure those concentrations? And we can measure it by mass concentrations, for instance, milligrams per liter. We can measure it as number concentration, particle number per liter, or surface area, meter squared per liter. And all of those can be important. Mass has always been the easiest to measure and what's traditionally been used for pollutants. Number concentration, we've developed in our labs TEM and AFM, transition electron microscopy and atomic force microscopy based methods. And we and others have developed SingleParticle ICP MS methods, which Dr. Merrifield will talk about in more detail. And for surface area methods, there are really no validated methods for nanoscale materials, but they may be important. And on the same slide, you can see data from a publication of ours where we looked at dose, or at least concentration within cells by a TEM method. And that's a useful method. And it gave us some invaluable data. But it was time consuming. It's only semi-quantitative. And there are other developments which need to be performed. And so the ICP MS becomes an invaluable additional tool. When we look at these different metrics, it's often assumed by the community that number and surface area are particularly important and relevant to nanoscience and nanotoxicology. But that's not always the case because, for instance, different processes which are relevant are dependent on different metrics. So, for instance, sedimentation may be mass-dependent, aggregation may be number-dependent, and contaminant absorption and uptake, so the absorption of metal or organic contaminants and delivery to an organism, may be surface area-dependent. So all of these different metrics may be important, but for our purposes, the number concentration is quite difficult to measure. In general, in the center and for some other groups, a multi-method approach has been used. And a way of illustrating the importance of this is really to think about the parable of elephants and blind men, which is fairly well known. And the story is about blind men trying to understand what an elephant is. And the one feels a leg of the elephant and thinks it's a tree trunk. Another feels the tail and thinks it's a snake, and so on. And that's what we're really trying to do with a multi-method approach, is to not look at individual aspects of the nanomaterial, but take a whole approach with our multi-method approach to understand the whole nano-elephant. And so, for instance, if you look at a familiar technique like dynamic light scattering, that is often biased to large aggregates. What you'll see by that method is large aggregates, while ignoring the fact that there may be dispersed particles or dissolved iron or other other shapes or precipitates of the original material. So by using a whole range of different techniques, we can measure a number of parameters because we're interested in things like composition, we're interested in size, we're interested in shape, and so on, but also reduce the bias so we can understand whether particles have aggregated, whether they're dispersed, whether they're dissolved, and so on, and also increase our information content of our samples before, during, and after exposure to organisms and to cells. And in terms of the single particle ICP MS, which I'll just talk about briefly, this is a hugely useful addition to a multi-method approach which we practice. We can measure a whole range of different properties of the particle with the single particle ICP MS. We can measure complex media transformations rapidly. And we can also increase our data by coupling with other instrumentation, such as field flow fractionation, which we're looking at currently. And with that, I'll hand over to Dr. Merrifield to talk about applications of the single particle ICP MS in more detail.
Thank you, Dr. Lead. I'm Dr. Merrifield. I'd like to talk to you about 3 applications that we have been working on in the center. Firstly, the transformations of silver nanoparticles in complex media. The second will be a complex mixture of nanoparticles using a combination of FFF and ICP-MS. And the third one will be looking at the uptake of contaminants such as ionic or nanoparticulate material into single cells. Firstly, I'd like to describe to some of you who are not as familiar with the technique how single particle ICP-MS works. Okay, so single particle ICP-MS is very similar to ICP-MS in that you have an introduction system that has a nebulizer and a spray chamber, and this introduces your liquid sample containing nanomaterials into the ICP-MS. We try and use a low concentration of nanoparticles of around 100,000 nanoparticles per mil so that only one nanoparticle will enter the plasma at a certain time. The particle enters the plasma, it will travel through the plasma, and it will heat up and burst and ionize. This will move through the mass selection process so we can take the element that we are interested in and go to the detector. When we get to the detector, we're going to use very short dwell time so that we can split the particle up into a number of different measurements. And at this point, we'll read it as an integrated intensity. And from this, from an ionic calibration, we can then go back and say how large our nanoparticle is of interest. The matrix of your samples can affect the ionization potential and the transport efficiency of your nanoparticles through this system, and I will discuss that in the next slide. So the first application I'd like to speak about is the transformations of nanoparticles in media. I'm going to go through a quick introduction on transformations of silver nanoparticles, some methodology, the importance of experimental design, and in this case using a core shell particle so that we have an internal standard already in our system, and measuring biometallic nanoparticles with single particle ICP-MS. And I'm going to speak about one result that we got today, which was looking at the concentration-dependent transformation of silver nanoparticles in moderately hard water. However, we have done a lot more of this study, which you can find in an application note already on Perkin-Elton's website. So the transformations of silver nanoparticles, as Dr. Lead mentioned earlier, we can have aggregation, where they'll stick together and sediment out from our suspension. We can get complex coatings, and as you mentioned earlier, having oxides, sulfides, chlorides, or natural organic matter coating, or we can have a process like dissolution. So it can dissolve, and from that you can have reprecipitation either on its own or with complexes, forming different shaped nanoparticles, or you can get an Oswald ripening effect where the particles that are still in suspension will grow from the dissolved material. The photograph at the bottom of this slide just shows silver nanoparticles that have been exposed to different conditions, and you can see they've gone from their usual yellow color to a rainbow effect. So we have some blue, green, and even red colored nanoparticles. So I'd like to talk about single particle ICP-MS and for core shell nanoparticles. In this study, the way we use them is by taking the gold signal and then taking the silver signal, so we'd get them separately. And when we measured the silver signal, we'll get a particle diameter and a particle number for the silver, and this silver will be allowed to react with the surrounding media. When we measure the gold signal, we'll get another particle diameter, which will be the gold core size, and we'll also get a gold particle number. The gold will act as the internal standard. So the gold is less reactive than the silver, but it's also protected by the silver layer. So really this shouldn't change. We should have the same particle number and the same particle size as long as nothing's happened to the whole nanoparticle system, like aggregation. So if the particle number for both gold and silver decreases, when the particle diameters for both gold and silver increase, then we're likely to be getting something like aggregation occurring. So experimental considerations for this. If we have a media composition, this will affect the instrument response. So increasing natural organic matter concentration will actually increase the number of particles measured. So we did a study where we did the ionic calibration and the nanoparticle transport efficiency in ultra-high pure water, and then added natural organic matter to the known amount of nanoparticle. And you can see as you add natural organic matter to your system, we end up actually getting more nanoparticles per mil. We haven't actually got more nanoparticles per mil, but we're changing the way that the instrument responds. So measuring the transport efficiency of nanoparticles in different media will give different trends. Transport efficiency for media containing salts will actually decrease. So if you look at the graph on the right-hand side, you have the ultra-high pure water, which comes out as a ratio of 1, because that's what we're normalizing to. And if you have moderately hard water that contains salts, we actually always get a slightly lower transport efficiency than you'd expect. And if you then add natural organic matter, we're getting a higher transport efficiency. So you have to make sure you matrix match your samples. So the same will happen with your ionic calibration. So it's very important to matrix match your standards with the test samples and do frequent quality control to check that you still have the same amount of expected nanoparticles in standard solutions. So I'm going to go through some results on concentration-dependent behavior and why this is important. So traditionally, when we look at transformations in medias, we're looking at around 50 to 1,000 ppb of your mass concentration of your contaminant. We go at these high levels because it's very difficult to actually look lower. The equipment that we used previously was unable to go lower in concentration than this. SingleParticle ICP MS has allowed us to go lower, so in this case we've also used 100,000 particles per mil and 10,000 particles per mil, which is 0.1 to 0.01 ppb of silver. And this is more in the range that we would expect to find in the environment. So SingleParticle ICP MS has actually allowed us to go to these environmentally relevant concentrations. So the media we used here was moderately hard water. We did a 48-hour exposure with a light-dark cycle of 16 to 8 hours at a temperature of 22 degrees. So if we look at the 44 million particles per mil solution, which are the graphs on the very left-hand side of this, we have a diameter and a particle number concentration. You can see here that the gold diameter increases slightly and the silver diameter increases slightly. So you've got to remember these are core shell particles. So with both of them increasing, we'd expect that we'd be getting aggregation, and then when you look at the particle number concentration, they decrease simultaneously. So we are actually getting what we consider aggregation in this case. Remember, this is the lowest concentration that's usually used, and most people think that we get aggregation at low concentrations of nanoparticles. If we then look at the two right-hand side graphs, you can see that in all the cases, the gold diameter stays very similar, and the gold particle number stays similar, but we're getting a decrease in the size for the silver particles and an increase in particle number for silver. This means that we're probably getting a dissolution from the main particles, but we're also getting a reprecipitation, probably with the salts in the media. So looking at the full set of results we've got a percentage change. So if you have a positive percentage change, we're getting a larger particle diameter. To the negative percentage change, we're getting a smaller particle diameter compared to the original. And the same with the particle number. If you're getting a negative change, we're getting less particles, and if we're getting a positive change, we're getting more particles. So I'd just like to reiterate the point that at the higher concentrations, we're getting what looks like aggregation, and when you go to lower concentrations, we're getting this dissolution and reprecipitation, which is something that we haven't really observed very easily before. So in conclusion, single-particle ICP MS in this case helps us to understand transformations in exposure media. The correct measurement for transport efficiency and ionic calibrations is incredibly important because you could get false positives very easily. The use of core shell nanomaterials makes it easier to understand these transformations. It would be very difficult to understand if we were just looking at the silver particles alone. And the environmentally and toxicological relevant conditions can be monitored so we can go down to very low particle number concentrations. We also showed in this study that nanoparticle to natural organic matter and nanoparticle to salt ratios were very key. So with that, I'd like to move on to the second application that we looked at. And here we were looking at the analysis of complex mixtures. So if we had bimetallic and monometallic particles in the same suspension, how can we tell the difference? Traditionally, this would be done with something like electron microscopy using EDS or STEM. Here we try to use FFF and single-particle ICP MS as a much faster way of measuring this. So single-particle ICP MS measures size distribution, and particle number concentration. We used a multi-method approach. So in this study, we used filled flow fractionation to separate our particles, and then which were mixtures of monometallic and bimetallic particles of the same diameters. And then we showed that this was a rapid technique compared to having to do TEM analysis. So first of all, I'd like to talk about separation for FFF and measurements of ICP-MS. So if we had a mixture of solutions that had monometallic particles of large and small within it, if we put it through the FFF, it's going to separate, so the smaller particles will come out elute faster and the larger particles will elute later. If you collect these separate fractions and put them through the single-particle ICP-MS, we're going to get a small particle for the first fraction and large particles for the later fraction, which will be synonymous to the size and diameter. So then if we looked at a bimetallic particle, say a core shell material, of a larger size, when those elute, they're only going to come out in the later fraction. But then when we measure with single-particle ICP-MS, we're going to see a much smaller size for the core and a large size for the shell. This is to do with the x to the 3 number of atoms per nanometer of particle as you go out in diameter. So then if we had a mixture of monometallic and bimetallic particles, these will all elute in the later fraction. And at this point, what we're going to end up with is 2 peaks coming in the gold signal, where we're having a small fraction from the core shell material and a large fraction from the monometallic material, and then we're going to see the silver peak also come out separately. So here we're looking at results, mixtures of monometallic and bimetallic nanoparticles. You can see that we've got on the left-hand side the gold signal, on the right-hand side the silver signal. The dotted black curves correspond to the expected signals from the monometallic particles, and the dashed lines are the expected signals from the bimetallic particle. You can see on the left-hand side that we're able to separate the monometallic particles from the bimetallic particles because of the size difference. And on the right-hand side, we're not actually able to see the difference between a large particle and the shell of the silver nanoparticle. So here's just some results that, one set of results that we have. Again, this is coming out as an application note soon, so there's more details in that. Here we're looking at suspensions of gold core silver shell nanoparticles, total of 80 nanometers. But here we're looking at a core of 50 nanometers and a shell of 15 nanometers and an 80 nanometer gold particle. We put 3 different ratios into the FFF and collected the fraction. So we have a 1:4, a 1:1, and a 4:1. On the left-hand side graph, we can see that we actually can measure the core size for the nanoparticle, the shell size for the silver nanoparticle, and the total size for the gold nanoparticle. The dark grey bars with an asterisk on top, they were actually calculated total diameters for the core shell particle from the known geometry of the particle. Then when we looked at the number concentration, of course the amount of core particles and the amount of shell particles were the same. So we could say that this is actually a core shell particle and not a 50 nanometer particle and a 60 nanometer silver particle. So we were able to actually calculate the exact number we had, and you can see that we came out with exactly the right fractions, a 20 to 80,000 particles per mil, a 50-50, and an 80 to 20. So we can actually get the right number of particles and the right size of particles in this case. So I'd like to conclude this and say we've shown that using separation techniques for FFF in conjunction with SingleParticle ICP MS, it's possible to measure complex mixtures of nanoparticles. SingleParticle ICP MS on fractions can give you a particle number concentration and a particle size for the particles within that fraction. And we can determine concentrations of nanoparticles in suspension for both the nanomaterials. The last thing I'd like to talk about is single-cell analysis and its environmental importance. So as Dr. Lead mentioned earlier, usually this is done in a mass metric. So here we're going to try and answer some questions. So do single cells take up ionic or nanoparticle contaminants? What is the exposure concentration? And in this case, we're going to use nanoparticles per mil. What is the dose? So this is how many algal cells in this case will take up nanoparticles or ionic contaminants, and how much is in each cell, and at what rate do they take these up. We used 3 cells of different sizes. They're freshwater algal cells. So the theory behind this is in A, you expose your cells to a metallic contaminant, either nanoparticulate or ionic, and then you move forward and you remove an aliquot of this and you wash it out and resuspend it in a media of fresh water. From this point, you can, in C, you introduce it to the plasma in the same way you do SingleParticle ICP MS, so you want one cell traveling through the plasma at a time. The cell will burst. The nanoparticles or ionic material inside it will also atomize and ionize. And then we'll go on to D, where you go back into the detector, and we're again using these very short dwell times so that in E we can get an integrated intensity per cell. So a cell will take up different amounts. If you're looking at an ionic contaminant, they're probably going to not be equal, so one cell will not take up the same as another cell. If you're looking at a nanoparticulate contaminant, you're probably going to end up with integers of the amount of that nanoparticle per cell. So I just want to show you some results, what you'd expect to see from the SingleParticle ICP MS in this. So this was a 3-hour exposure, and obviously we did a cell control and an ionic control. In both of these, after 3 hours, we didn't see any nanoparticles as you'd expect. However, if we look at C, where we had cell plus 1 ppb of AU ionic, we actually ended up seeing that we're getting some cell coming through with a certain amount of ionic within them. So you're seeing that there's an uptake from some cells. And then if we look at D, where we had cells plus gold nanoparticles, in this case they were 60 nanometer NIS, we actually exposed to around 1 ppb again, which is about 500,000 cells per mil, nanoparticles per mil, and we had cells at about 1,000 cells per milliliter. And you can see that we're getting a nice even distribution as you would expect from a NIS nanoparticle, so these cells have actually taken up nanoparticles. So if we actually look at some results, the things that are interesting that you can get from here, here we're looking at total number of cells, which were measured by hemophotometer in black, and then metal-containing cells, which are the grey bars. So in our 3 different species, we're getting 3 different responses. So our small algal cell, which is the chromis FT, we can see that we actually counted around 100% of the cells in each point after 3 hours of exposure. So we're not really getting much cell death or cell growth in this period of time, which is what you'd kind of expect. If you look at the cell control and the ionic control, we're not getting anything coming through in terms of percentage of cells containing any metals, which is again what you'd expect. We didn't add any contaminant and we didn't have any nanoparticles. When you go through to the cell plus ionic and the cell plus nanoparticles, you can see around 50% of our cells actually took up either some ionic material or some nanoparticulate material. This is actually in contrast to the other 2 cell species. So the one on the right-hand side, which is our middle-sized cell, only took up about 20% ionic and nanoparticulate material. 20% of the cells took any of this stuff up. And then if you look at our large cells at the bottom, which are about 60 micron cells, you can see that, again, both the cells took up ionic and nanoparticulate, but much more of the nanoparticulate material is taken up than the ionic material. So here we can show that uptake of material is dependent on cell strain and contaminant type. You can also measure the ionic concentration simultaneously. So here I just wanted to show you a quick graph to show you why this is important over time. So our cell control had no ionic material in it, nor did our cells plus nanoparticles. Our ionic control had 1 ppb in, which is 100% of the material. Our cell control after 3 hours, so it reduced, went down to about 60% of our original amount of ionic material. So we lost 40% of our ionic material. This means over time, our exposure is actually changing because of uptake from the other cells. So the kind of interesting stuff we can get from this is just a table to show an overview of the results that we got. So I've shown that you can look at the exposure, which is the part on the left-hand side. We added an initial concentration that was measured in all samples to be around 0.94 ppb of gold. And you can see after 3 hours that we lost anywhere between 40 and 60% of the initial added amount of ionic material. And you can see the same with the particle number concentration. We added around 500,000 particles per mil, and you can see that after 3 hours, different amounts of these particles have been lost to the system. And then on the right-hand side, we can now talk about dose. So you can measure your number of cells at the beginning and at a 3-hour time point, and you can say how many cells you have. And then you can say from the single cell ICP MS data, the percentage of cells that contain metals. So you can see that the smaller cell contained around 50% for both ionic and nanoparticulate. And then when you go to the larger cell, 15% of the cells contained ionic, but 87% of the cells contained a nanoparticle of some description that was measured. So then you can actually go and look at your data. So we had a nanoparticle size for each of these uptakes. So we had like a 22 nanometer, or what can be considered a 22 nanometer size particle contained within the ionic exposure for the first cell. And you can see that you have a particle size of around 55, when you expose it to nanoparticles. So now we've got the percentage of cells that have taken something up, and we know how much each cell has taken up, which can be transferred into a mass concentration. I'd like to point out that this is in attograms, which is 10 to the minus 18 grams per mil. And you can see that we're getting anywhere between 60 and 940 attograms per cell. So now we know the percentage of cells taking it up and how much material has been taken up from each cell, which is going to be very useful if we're looking at exposure and dose concentrations, especially in medical applications. So the conclusion to this technique allows us for an accurate measurement of exposure and dose on a single cell basis and in metrics that we actually want to measure in, which is nanoparticles per mil and cell amount taken up per cell. You can independently measure a biological response. In this case, we measured cell growth, but you could measure DNA damage or a biological assay to give you your biological response. And then we've shown that algal cells take up both ionic and nanoparticulate forms of contaminants, but the uptake is cell dependent. It's dependent on the strain that you have. So with that, I'd like to thank everybody for listening. It's the end of the presentation, and I'd like to invite any questions.
Thank you very much to the three of you for that excellent talk. It's now time for questions from the audience, and I would just like to remind the audience before we get started how to do that. If you'd like to submit a question, just type it in the Q&A box, which you can find on the right-hand side of your window. Okay, so let's look at our first question here. Dr. Lee, this one I think is probably for you. So should we be concerned about nanoparticles and the environment or human health?
It's a difficult question to give a exact answer to. If I refer back to my talk, nanomaterials are a wide range of different types of materials. And so the question is similar or analogous to saying, should we be concerned about chemicals? And certainly it looks like some of them are a cause for concern. So we've been talking a lot in this talk about silver, and silver is used because it's of its known toxicity, particularly to bacteria, to algae, to other life forms in the environment. The toxicity to humans seems to be much lower, and the exposure concentrations are such, particularly in the environment, are such that it may be a cause for concern, so that the exposure concentrations may be high enough to cause hazard to bacterial, to algal and other important species in the environment. Whereas gold, the other material that we talked about in some detail, is thought to be lower in toxicity and probably lower in exposure as well. So therefore the risk is significantly lower. So I think there is cause for some concern, but there are also huge benefits in using these materials for environmental and other applications. And so we have to consider the benefits and the risks at the same time. And to a large extent, it's difficult to answer that question other than on a case-by-case basis for particular particles. But it certainly seems it's something that perhaps shouldn't necessarily stop us innovating and using these particles, but it's something that we need to be concerned about to ensure that there is no significant hazard and risk.
So then how does the information that we gain from using single-particle ICP MS improve our knowledge about such toxicity or potential toxicity?
So in particular, it offers a whole new way of looking at information. So as I said, it's a very valuable addition to a multi-method approach, but it does give us some unique things which we can't really do in other cases. So particularly looking at low and relevant exposure concentrations in the environment and other systems can't be done easily or at all with a lot of other analytical techniques. So previously, as in Dr. Merrifield's talk, we've been looking at concentrations which are in several micrograms per liter up to milligram per liter, which are much higher than real exposures. And ICP-MS, single-particle ICP-MS, gives us this ability to look down into these realistic concentrations and then relate those concentrations and also size measurements and other parameters to biological responses, so we can get some understanding of how dose and response relate to each other under realistic conditions.
Excellent, thank you. This next question I think is for Dr. Merrifield. What is involved with sample preparation for these methods? Do you have to acidify your sample?
No, so for single-particle ICP MS, you don't really want to acidify your samples. If you acidify your samples, you're going to turn your nanoparticles into an ionic form, and then you're not going to have anything to measure. So generally, we don't acidify our samples because you don't want to break up your particles. Excellent,
thank you. And Dr. Merrifield, do you see a membrane effect from the FFF, and is there any carryover?
So there is a membrane effect with FFF. In this case, what we did is we negated it slightly by using actually higher concentrations in the FFF and having to dilute a lot to go through the ICP-MS, single-particle ICP-MS. But if you couple them straight through, then yes, at the moment we'll see membrane effects.
Excellent. One more for you, Dr. Merrifield. On slide 30, it looks like the 80 nanometer gold particle has a bimodal distribution. Is that the case?
Okay, so this is a slide I think everybody's talking about. So yeah, if you look at the left-hand side graph, it looks like we have a bimodal distribution for the 80 nanometer particle, gold particle. In this case, we actually bought these particles in from a supplier rather than making them ourselves. And when we actually did get them, they did have a bimodal distribution. So, I mean, it didn't matter for the experiment that we were running. We could still separate these particles, which shows how good single-particle ICP-MS is. And it also shows that you can get degradation from particles, and it's very difficult for manufacturers to get monodispersed particles on a regular basis. So this is actually an easy technique that you can use to check your particles when you get them in to make sure that they are monodispersed and not bimodal. So yeah, in this case, it is bimodal, and we didn't really worry about it.
Thank you. Dr. Lead, why are you interested in number concentration and not mass concentration for nanosamples?
Well, number concentration is an important metric for nanoparticles because it tells us a lot more about this interaction or this potential interaction with particles and with organisms. And so, for instance, one of the important transformation mechanisms is aggregation from small particles to large particles, and as I showed earlier, that has potentially a great effect on toxicity and certainly on environmental behavior. But that transformation process from dispersed individual particles to larger aggregates or agglomerates is proportional to the concentration of the individual starting particles, so the number concentration. So it gives us a handle on understanding these kind of processes and being able to quantify these processes, which have so much effect on environmental and biological effects.
Thank you. Dr. Merrifield, when you're looking at the cell uptake, how do you know the cells are making it to the plasma as single cells?
Okay, so we actually ran a cell viability test before doing this. So we wanted to make sure that the cells are making it through the nebulizer, because the nebulizer is where the most pressure is going to be on the cell and the point where it's most likely to burst. So we had to make sure that we were actually getting cells through the nebulizer as whole cells, whether they were alive or dead. So we did a cell viability test at different neb gas flows to check that we were actually getting all the cells through to the nebulizer, so they could into the spray chamber, so they could actually pass through into the plasma.
And how does information acquired by the single cell analysis compare to traditional methods for analyzing nanoparticle uptake, such as fluorescence or electron microscopy?
Okay, so fluorescence and electron microscopy, I've done a lot of electron microscopy in the past looking for nanoparticles in single cells. And in both those techniques, it's really a semi-quantitative or a qualitative analysis. You end up having to spin your cells down, and in terms of electron microscopy, spin your cells down. It takes like a week for you to turn them into a resin and slice them, and then the actual test that we did earlier, that was shown earlier with the serum dioxide nanoparticles into cells, that actually took me probably a month of nights to image the amount those slides. So, and we still only got a semi-quantitative result from it. So in terms of time, it's a lot faster. It takes like 1 minute to do the analysis in single cell ICP MS. And the sample preparation is much quicker and much less stress on the cells. And also, you actually get quantitative information out of this, so we can actually say the percentage of cells that contain metals and how much metal per cell. So before you would digest, say, 5 million cells, and say you had 1 ppb of contaminant in them, but you don't know which cells are taking them up and how much has been taken up per cell. So this actually really extends the knowledge that we can use for this.
Fantastic. Dr. Lead, going back to the effects of the nanoparticles, are there any differences in how you treat toxicology, ecotoxicology, and environmental samples?
That really depends on the individual cases, I think, the individual studies. And so there are few, if any, differences between those three areas of toxicology, ecotoxicology, and environmental samples. But it does, and there is considerable overlap between them. So, for instance, the synthetic waters, environmental waters that are used and that were talked about during the webinar can be used as exposure media for ecotoxicology. And in terms of analysis and dose response and so on, the differences really come into play in terms of the nature of the nanoparticle and the nature of the exposure media. So the particle size and coatings, particularly in composition, can have a large effect on how the particles behave and transform in particular exposure media. What you tend to find in terms of their properties is that salts will often cause aggregation, whereas natural organic matter and proteins will coat nanoparticles and change their surface behavior, often protecting them, but also often changing them into other materials. So there are similarities between environmental and human studies in that natural organic matter and protein can have similar behaviors in terms of the nanoparticle processes and properties. So there are no distinct differences between them, and in fact there are a lot of similarities, but it really depends on looking at the very specific nature of the study involved, the nanoparticle type and the media composition particularly.
So then, are there any current regulations, environmental or health regulations, that govern nanotechnologies, whether it be using them, selling them, or importing them?
Yes, there are a lot of regulations that are linked to or involved with nanomaterials in North America, in Europe, Australia, and elsewhere. And one thing perhaps missing is one thing about the specificity towards nanomaterials. So there are frequently no considerations of nanospecific properties in these regulations. We haven't reached that step, but there are a lot of regulations in a number of countries which deal with these from an environmental and a safety perspective.
Excellent. Let's just take one last question. Actually, Dr. Steffen, this one would be for you. So it looks like single-particle ICP MS applications are continuing to grow. What do you see coming down the line?
Single-particle ICP MS applications are growing indeed. We've been applying it to a lot of environmental samples, and now we are, as Dr. Merrifield was demonstrating, was talking about in her section about the use of the similar data interpretation, but instead of applying it to a nanoparticle, we're applying it to a single individual cell. With the Dr. Merrifield experiment, we focused on algae. Now we are trying to look at different cell species, cancer cells, yeast cells, bacteria cells, trying to incorporate the same level of data interpretation of looking at what we call a discrete signal coming from a burst of an ion plume generated suddenly in the plasma. So the application of single-particle ICP MS are growing. They're migrating into the biological field. On the other hand, we're using single particle to understand chemical processes, in which we are looking at how a low-level contaminant, they are originally at the ionic level, but for some chemistry happening in that process, chemical process, they might change in form to generate some clusters, some colloidal form. And single particle ICP MS is opening up few application areas in that field as well.
Fantastic. Well, we're out of time and we need to wrap up. So I'd like to thank the three of you for your talks today and for answering all those questions. I'd also like to thank the audience for participating today, and I'd like to thank our sponsor, PerkinElmer, for making today's webcast possible. Today's webcast will be available for on-demand viewing through April of next year. You'll receive an email from Spectroscopy alerting you when the webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.